Okay, this is section 6 of the material for this week. I'm now gonna move on to the second set of PowerPoint slides up on Blackboard. L15, Chapter 20, Resistance. So this is looking at resistance to antimicrobial medications, which starts on page 515 of the textbook. First of all, we're going to look at the mechanisms of acquired resistance. So how do the bacteria actually become resistant? Not how they become resistant, how they beat the drugs. And number 2, we'll look at how they become resistant. What's the mechanism that allows them to beat those drugs? It's a bit of a fine line there, but hopefully we'll get that sorted. So, slide 2 in this set of slides, resistance to antimicrobial medications. When penicillin G first came out many decades ago, it was used to treat, amongst other things, Staphylococcus aureus. And when it first came out, only a small number, about 3% of Staph aureus were resistant to penicillin G. Now, why were any resistant in the first place? So remember where, or indeed think about where penicillin came from. It came from a fungi. So bacteria and fungi and bacteria and other bacteria, all kinds of organisms, have been living together in the environment and fighting each other for hundreds of millions of years. So humans weren't the first organisms to ever try penicillin against bacteria. However, since we have been using antibiotics in such large amounts over those last few decades, now if you have Staph aureus, it's very likely that it is resistant to penicillin G. Now more than 90% are resistant. So you may be familiar with the idea of the growing importance of antimicrobial resistant bacteria. And we're going to really focus on the bacteria this time, as in these previous sections I've been focusing on bacteria too. And it's no surprise that bacteria are becoming resistant just from the huge quantity of antibiotics that are used in the environment every year. Hundreds of tons used per year. We'll look at slide 3. We'll look at the idea of mechanisms of acquired resistance, and we'll look at how they get that resistance. 2 general ways: through spontaneous mutation and through the introduction of genes via things like plasmids or viruses. Let's move on to slide 4, which is the mechanisms of acquired resistance. Actually, let's go back to slide 3, because I guess we need to talk about the difference between acquired resistance to innate resistance. So innate resistance is something that the bacteria has already. For example, gram-negative bacteria, they have an innate resistance compared to gram-positives because they have that outer membrane, and that outer membrane gives them an additional barrier for the entry of antibiotics to the cell. Also, a microbe like Mycobacterium tuberculosis, that has a lot of innate resistance. It's innately resistant because it has that unusual waxy cell wall. It has innate resistance because it has this very slow growth rate. So these are innate resistances that just are found in these different kinds of bacteria. Acquired resistances are more like the bacteria doesn't have them, and then it does have them. So a gram-negative has the outer membrane that gives it innate resistance, and then on top of that it may acquire additional resistance, say through a plasmid. Or a mycobacterium has those factors that make it innately resistant, but on top of those it might acquire some genes through mutation. So if you're going to acquire resistances, these are things that the bacteria can get to help it to fight off drugs. On slide 4, we have number 1, antibiotic inactivating enzymes. Okay, so antibiotic inactivating enzymes, we've seen these in the previous sections. We have our penicillin binding proteins, their job is to bind a substrate and then go ahead and build cell walls. Sorry, I had a glare on my board there. So, what we do is that we have our drug, and that drug mimics the shape of the substrate. The drug binds the active site of this penicillin binding protein and prevents it from building the walls. So the bacteria fights back with a beta-lactamase that snips up the drug. The drug can't fit into the active site anymore after it's been snipped up. And our bacteria is then once again free to use its enzymes to build those cell walls. There are other examples too. We looked at the idea of shape matching in ribosomes. So here's our ribosome, our 2 subunits. Here's that binding site in the 50S subunit. A drug such as chloramphenicol might come in and bind to that site, and then it would interfere with protein production. But if the bacteria can make an enzyme that interferes with the drug, maybe changes that drug's shape, that drug can no longer fit into the binding site on the ribosome, and if the drug can't fit, it can't interfere. So, enzymes that inactivate drugs is number 1. Alteration of the target molecule is number 2. And those two same examples hold true. Is our penicillin binding protein once again. Here is our drug. It's going to fit into that binding site, but the penicillin binding protein changes the shape of its binding site. And now the drug is no longer a good fit. The drug no longer fits. If it doesn't fit, it doesn't block. Clearly, if any changes happen to the shape of our penicillin binding protein, it still must function to bind its substrate and use that substrate, in the case of PVPs, to build walls. So through changing the shape of the penicillin binding protein, it can still work with its substrate to build the walls, but it will no longer fit the drug. Back again to our 50S subunit. There's that binding site for our drug. Change the shape of that binding site. The drug doesn't fit. If the drug doesn't fit, then it won't have any effect. Number 3 is decreased uptake. This is particularly true for gram negatives. There's their cytoplasmic membrane, there's their cell wall, and here is their outer membrane. And the way that things move backwards and forwards across the outer membrane are via protein channels. So let's imagine you were a bacteria in an environment where antibiotics were an important factor. There's going to be a range of bacteria in that environment. Some are going to have 20 channels. Now this is just a number I'm making up. One bacteria will have 10 channels. One bacteria will have 3 channels. In an environment where there's antibiotics, having fewer channels is probably an advantage, and therefore those bacteria with fewer channels will be more successful, and they will pass on, or indeed they will have offspring that have fewer channels. So that's how a bacteria might adapt to a antibiotic environment, by selecting the bacteria that have the fewer channels and therefore reducing the amount of drug that gets into the cell in the first place. So reduction in number of channels is important. Reduction in the change in the configuration of the proteins in that channel. If the channel is a protein, if that protein changes its shape, it can then prevent molecules from passing through that channel. So you can see the 3 outcomes on slide 6. You may have loss of these channels called porins. Those protein channels have to be put into the membrane to begin with. And there's a whole lot of different things that could happen to just simply prevent the porins being put in the membrane. Second one, we can have a configurational change of the channel that doesn't allow certain molecules to get in, whereas beforehand those molecules could get in. Or we could just have a decrease in number of channels. There's 2 channels in that section in the wild type, but only 1 channel now in the bacteria that's adapting to this antibiotic environment. So the first thing that a bacteria might want to do in terms of its channels is to stop stuff getting in. Stop stuff getting in either by reducing the number of channels or changing the way that the channels work. The other important thing, number 4, is that some bacteria have pumps in their channels. These pumps are called efflux pumps, and they do what they sound like. Anything that gets into the membrane is just pumped out again. This is a very kind of troubling mechanism for resistance because it's so general. You don't need any fancy shape matching or alterations. You just put in a pump, and anything that gets in, pumped out, and the drug doesn't have any chance. And it doesn't matter what drug it is, whether this is a beta-lactam drug, it's pumped out, or whether this is a macrolide, it's pumped out. Normally, you would have to have 1 change to counter the beta-lactam drug, another change to counter the macrolide. Here, 1 change counters both of those drugs. Pseudomonas aeruginosa, sorry. These are those bacteria I've mentioned before that are capable of living in disinfecting solutions. And one of the main mechanisms they have with being so resistant is the presence of these efflux pumps. And there's the chance that the pump mechanism, if 2 bacteria get together, they can get together of different species. Maybe the Pseudomonas can start giving its pumps away to other kinds of bacteria. So that was looking at what the bacteria can do to make themselves resistant, i.e. the mechanisms by which they can combat the drugs. And now this next section, slide 7 and 8 and 9, these are looking at how those changes actually occur within the bacteria. So the first thing that can occur is that we can have mutations. Mutations are changes in DNA. Changes in DNA sequence in A's, T's, G's and C's. So we have genes, messenger RNA, we have proteins, we have transcription and translation. A gene is transcribed, and that is then translated into a protein, and the sequence of the gene describes the sequence of codons in the messenger RNA, and the sequence of codons describes the sequence of amino acids, and the sequence of amino acids describes how that DNA is going to fold up. You change, say, a letter in the gene sequence, you change a codon in the messenger RNA, you change an amino acid in the protein, and now you have a protein that folds up in a different way to the protein as intended. And you can see that an example of that on the slide, where a ribosome changes its shape. It is folded up so that the drug can fit in. but then it changes, and that now the drug can't fit in. A letter change in the gene that built this section of the ribosome. That letter change causes the protein to fold a little different, and now when the drug could fit in, it can now no longer fit in. And the drug can't fit in, therefore it can't stick and it can't have a negative effect. So on to slide 8. In a typical bacterial infection, we are dealing with a lot of individuals. And mutations in letters of DNA are random. But if there are a billion cells, it's likely that many of those bacteria have different mutations. And that's where we get our variation from. So the variation in the shape of the large subunit of a ribosome. Lots of different bacteria, all with different slight variations in that shape, because they all have slight variations in their gene sequences. And those different variations in ribosome shape are going to interact in different ways with the drug. And in a high drug environment, those shapes that prevent the drug from affecting the cell are beneficial, and they are selected for. Those shapes that allow the drug to fit, they're going to either kill the bacteria or they're going to prevent it growing, and they'll be selected out. So this is a process of natural selection. The mutation is the underlying basis of the variation in penicillin binding proteins, variation in ribosome shape. Those shapes are selected for in terms of which bacteria can survive the best. The one that can survive the best passes on its genes to the next generation. It passes then on the mutation. So a bacteria that has gained a mutation making it resistant, its offspring will have that mutation too, and they will also be resistant. And then we'll have another round of natural selection in the next resistant population. They'll have variation, and some of those will be even more resistant. And that's how we arrive. We start off with a large number of bacteria with an amount of variation, and generation after generation we select those bacteria that are best able to survive. And if we are bombarding the bacteria with antibiotics throughout that entire time, we are selecting for then bacteria that can resist those antibiotics. Slide number 9 looks at the other important way by which bacteria can gain resistance. They can be given resistance through this thing called a plasmid. One bacteria has its own circular chromosome, and many bacteria have, in addition, a smaller circular piece of DNA called a plasmid. These plasmids can contain, or indeed do contain, genes, and those genes may be important in building something, like a beta-lactamase. So this bacteria with its plasmid, it can build a beta-lactamase. It's resistant. This bacteria without the plasmid can't build the beta-lactamase. It is susceptible. But this bacteria and this bacteria can get together through a process called conjugation. This bacteria can replicate its plasmid, and it can give it to its neighbor. And now suddenly this new bacteria has the same resistance as the old bacteria. And it turns out that bacteria aren't particularly careful about who they give their plasmids to. They can get together with lots and lots of different kinds of bacteria, not just their own species, and they can have this free exchange of plasmids. So it doesn't take a whole lot for a plasmid in a population of bacteria that are being bombarded with antibiotics for the bacteria that has it to start giving it to everybody else. And then the genes on this plasmid will be transcribed and translated into proteins. Maybe one of those proteins will be an important factor in resistance. Typically, if a plasmid contains resistance genes, it is known as an R plasmid. R for resistance. And this process of giving plasmids to your neighbors is known as horizontal gene transfer. Because we can have a bacteria that has offspring, and that bacteria can pass on its DNA to its offspring. That is vertical gene transfer. If a bacteria gives a plasmid to a neighboring cell, this is our horizontal gene transfer. Horizontal between unrelated bacteria, vertical from parents to offsprings. So slide 10 has all that stuff on our study guide: acquired resistance, antibiotic inactivating enzymes, alteration to target molecules, decreased uptake, efflux pumps. Just understand how all those mechanisms work, and then how do our bacteria get those mechanisms to begin with. They can go through selection via spontaneous mutations that are selected for in the population, or they can start to give away their resistance plasmids. And if a plasmid is important in resistance, then it's called an R plasmid. Okay, that's that for part 6.